In today's lesson, we're going to be looking at meiosis and how gametes are produced. So one of the questions that we need to be asking ourselves is, why is the production of gametes called meiosis, whereas the production of other somatic cells called mitosis, and how is that different? And just to start you off, I've put this animation up of the stages of mitosis just to remind you what's happening here. So we have DNA replication, then we have prophase, metaphase, anaphase, and telophase, resulting in cytokinesis, which is happening now. And as a result, we have two identical daughter cells with the exact same number of chromosomes as the original. Learning objectives associated with today's lesson are, as you can see on the screen. So it's a lot of define and explain and distinguish in the first three. Now, to distinguish means to tell the difference between, and for those higher order questions we want to be able to explain why mitosis and meiosis are different. So that was the learning question at the start, and we need to be able to list and describe how meiosis occurs and the order. These are the key words associated with today's lesson. Some of them may seem familiar to you, others may require a bit of reading of the textbook. Okay, so before we move on, we're just going to revise some of the terminology we've seen before. So we know that the normal human chromosome complement is 46 chromosomes. And these chromosomes exist as 23 pairs. And the 23 pairs that we get, we have 22 autosomes, those are body chromosomes, and one pair of sex chromosomes. Now I've written that they're matching here, but with genetic males it's XY, and it's not strictly true, but just for simplicity we say we have one pair of matching sex chromosomes. So following on, looking at sex chromosomes, genetic males will have one X that they inherit from their mother and one Y chromosome that they inherit from their father, whereas genetic females will inherit the X chromosome from male and female parents. Now we know that the matching pairs of chromosomes that we get from each parent are known as homologous pairs. So therefore, that means that we have males with 22 homologous pairs, whereas females have 23 true homologous pairs, because the X chromosomes are matching in every way. Now these diagrams are called karyotypes or karyograms, and they illustrate what we've just been talking about. So in this karyogram of a normal genetic male, we can see that there are two chromosomes at position number 1. One would have been inherited from the male parent and one from the female parent. Now they are described as homologous pairs because they both carry the genetic information carried on chromosome 1, and they will have the corresponding genes at the same position on each chromosome. We can see that the last pair of chromosomes, the sex chromosomes here, denotes the genetic sex of the individual, and we can see that this person is a genetic male and they have one longer X chromosome and a shorter Y, which isn't strictly homologous in every way, which is why we say that males have 22 homologous pairs. If we look at the female here, we can see again all the homologous chromosomes all lined up, and at the end the sex chromosomes on this normal genetic female, they match up exactly. Now a quick summary of the terms haploid and diploid. When we talk about diploid cells, we are referring to cells that contain the full normal chromosome complement of 46 single chromosomes or 23 pairs of chromosomes. Diploid cells are referred to as having the 2n number. So that means 2 times n, in this case n for humans is 23. So 2 times 23, which gives us 46. Now normal body cells, also known as somatic cells, contain the 46 diploid number of chromosomes, as does a zygote. Now if we look at the gametes, which are sex cells, and in the case of humans that would be a sperm and ovum or egg, these cells are very special. They contain 23 chromosomes, which is half the number of body cells, so half the number of normal somatic cells. Therefore, they are haploid. So gametes are haploid, and they're simply given the N number, N standing for 23, remember. Now, when we look at the image of how the cell flow works, we start off with gametes undergoing the process of meiosis, and this occurs in the gonads. So we'll talk more about that in the next unit, but gonads in females are the ovaries, because those are cells that produce gametes, and in males they are the testes. So we end up with 23 chromosomes in each of the gametes produced by the ovaries and the testes. This occurs through the process of meiosis. Now, the egg and the sperm, when they come together at fertilization, we will then end up with a zygote. Now, the zygote has 46 chromosomes, or the 2N number, because the chromosomes found in the egg and the sperm fuse. So that's 2 times 23, which gives us 46. Now, as the zygote grows, it will undergo rapid mitosis, as we discussed in the last lesson. So the somatic cells will grow and divide rapidly, and all of those cells will become diploid cells. Now, as these diploid cells mature and differentiate to become specialized cells in various organs of the body, we quickly find that they assemble as cells become tissues, and tissues become organs, and then organ systems, and so on. And then we end up with fully grown humans, and then the life cycle occurs again. Okay, so now we're going to be looking at the significance of the haploid number of cells. Now, gametes, which are produced at the end of the entire process of meiosis, they must be haploid, which is the N number. Again, in humans, that's 23. Now, if that didn't happen, the normal chromosome complement would not be achieved at fertilization, which we know is 46 chromosomes. And that can actually result in genetic abnormalities, which we will discuss, or it could actually result in failure to produce viable offspring. And what that means is that you wouldn't have babies that are born, which would significantly affect the human population. Now, here in this diagram, just got an image to show these cells here are what we would call gamete mother cells. So these cells contain the full chromosome complement. In this simple example, those are 2 chromosomes. So therefore, during meiosis, we produce haploid gametes. So they contain half of 2, which is 1, which is found in each gamete there. When these gametes meet at fertilization, we end up with the diploid number. So the zygote has a diploid number of chromosomes, and that can go on to produce new diploid cells and hence produce a new organism. Okay, so now we're going to start talking about meiosis proper. Now, even though this diagram looks a bit busy, I'm just going to take a moment to look at different parts of it. So the first thing we're looking at is interphase. So remembering back to the cell cycle, the majority of the cell cycle is actually interphase, and we know this is where DNA replication occurs. So this is when DNA, instead of existing as one single chromosome, it replicates itself, and we have one double chromosome. Now, in this particular cell here, we can see that there are 4 chromosomes, so the diploid number is 4. Now, the difference between mitosis and meiosis we can very quickly establish is that instead of 1 round of division, we have 2. Now, what's important to understand about meiosis is that we must go from having the diploid number of of chromosomes in each cell to the haploid number. So if we look here at the start, there are 4 chromosomes, and then at the end, each cell has only 2. So this is what we want to achieve in meiosis. Remembering, meiosis only occurs in the production of gametes, which are sex cells. So here we have prophase 1, so this is the first division, metaphase 1, anaphase 1, and telophase 1. Now at the end of that telophase, we can see that even though we have 2 chromosomes in each cell, the issue is they're double chromosomes. We can't have that. They need to exist as singles, otherwise it's too much genetic information. So that's why meiosis occurs twice. And you can see that each of these 2 cells that were produced at the end of meiosis 1 will run through that exact same process. And as a result, at the end of that, we have 4 haploid cells. Okay, so there are some specific things in meiosis that we need to be aware of. So first of all, we understand that meiosis is a cell division in which the number of chromosomes in a gamete mother cell, so a gamete mother cell is a cell that produces gametes, and remember gametes are egg and sperm. So meiosis is when the number of chromosomes in gamete mother cells are reduced from 2 sets, which are diploid, to a single set, which is haploid. Now you can see that again here. So at the top in this example, you have got 2 chromosomes in the gamete mother cell, and down at the very bottom, in the resulting gametes, we have 1 chromosome in each. So now we're going to look at how meiosis works. So again, we're going to use the same simple diagram purely because it's only got 2 chromosomes, so it's easier to follow. So first of all, we understand that chromosomes exist as single threads of information. We know it's tightly coiled. We understand that it exists as chromatin, which is supercoiled. Now we know that before meiosis occurs, DNA replication will happen in interphase. Now what's very different here between meiosis and mitosis, the first point of difference is that we want to increase genetic variation. So that's really, really important. This is how we get lots of different people on the planet. So genetic variation can be achieved, first of all, through portions of chromosomes switching with each other. So if we see here at the top, we've got single chromosomes. They will double and become double chromosomes through DNA replication. And then in this diagram here, we can see that those double chromosomes have actually crossed over each other. And as a result of that crossing over, they have switched portions of genetic material. Now if we follow that down in the red chromosome line here, we can see that these 2 chromosomes in the resulting gametes are no longer identical. So this one is purely red, this one has a portion of the blue in it. So this can explain why you may look really like one parent but have features of another. Now what's really important to understand here is that this exact point at which that crossing over and switching of genetic information occurs is called chiasmata. Now, in the second meiotic division, there is no more chiasmata, okay? But we do get other things that enhance variation. So again, the chromosomes line up at the equator. During metaphase, they are pulled to opposite poles. Again, this is similar to mitosis. This is anaphase. And as a result, we have telophase, which is a nuclear division, and then cytokinesis. Now, at the end of the second meiotic division, we have 4 genetically unique daughter cells, and they are all haploid. Now, I'm going to go back and revisit this idea of crossing over. So up here, remembering this only happens during the first meiotic division, really important. this double chromosome, let's say from the maternal parent, and this double chromosome from the paternal parent, they are switching. So at this point here you have crossing over happening. And what's occurred is that segments of genetic information have been swapped. Now it's the same information but different alleles. So you can see that that has been given by the different letters. And again, the point of crossing over is called chiasmata. And I'm showing you that here with the yellow arrow. Now, why is chiasmata important? Again, we've discussed this. It's because it increases genetic variation in gametes. If all offspring looked exactly the same, they would all be wiped out by the same diseases. So by having variation in a population, it increases the chances of survival. Another instance in which genetic variation can be achieved, in addition to crossing over, is through independent assortment. So this is when the chromosomes line up along the middle of the equator at metaphase. They line up in a random order. We're not sure the order they'll line up in. So if you think about school children lining up for their lesson after recess, every day they do that. They do that independently of each other. There's not a big amount of thought that goes into that. It's random assortment. So whoever lines up first, second, third, fourth, and fifth, and so on, is going to be different sequence each time. And that the same kind of eye principle can be applied to independent assortment of chromosomes. So if we look at independent assortment, the random alignment of homologous pairs of chromosomes is what independent assortment is. Now, in this example here I'm showing you from the top, you can see that you have got, it just so happens that all the dominant genes with the capital letters line up on this side of the equator, and all the recessive genes line up on the other side. So you don't have a mix of dominant and recessive genes. So therefore, when we have meiosis, you will have some gametes with only dominant genes and some gametes with only recessive genes. So if that egg was fertilized by a sperm, you might end up with an individual with very dominant dark features if we're talking about skin and hair color. In the second picture, we can see that we have got a mixture of dominant and recessive genes. And what that means is you may have, for example, blue eyes, which is a recessive trait, and really dark brown hair, which is a dominant trait. So this is how you get that mix of genetic information. Conversely, down here, you might end up with dark eyes and blonde hair, if we're using those similar examples. So what this is showing us is that independent assortment of chromosomes, so the way that they line up along the equator in metaphase and dictating which cell they'll go into at the end of meiosis 2, will ensure genetic variation. So again, it's just another diagram to illustrate that point of independent assortment. Okay, so here's a video that describes the process of meiosis. It's quite a good one. It's only 3.5 minutes. I would really recommend that you watch that. Okay, so now that we've talked about meiosis and mitosis, what I would like you to do is to make a table to compare the differences. Now, this is really important because this could be an extended response question or a short answer question that could be worth maybe 6 marks. So being able to write and create your own comparison table is a really easy and neat way of getting down those facts in a presentable way that your marker will appreciate. Once you've done that, I would like you to complete the notes on pages 16 to 21 of your class booklet. Definitely want you to read pages 254 to 261 of your textbook to help you writing good quality notes. And please consolidate your learning on meiosis using pages 272 to 273 of your textbook, and complete questions 11 through to 15, and 18.